Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Hi, and welcome back to Scigest. My name is Plunkett, one of the hosts here, and today we have Dr. Revel Drummond joining us. Hi Revel, thanks for having some time today.
Hi, and good morning.
So Revel's an expert in gene technologies, and we've asked him to join us today to discuss the landscape in this area. So we're planning to do a two-part series. First, today to go over some of the terms and definitions used in the genetic technology area and to get a bit of a background. And then next time we hope to go a bit deeper into the history, the modern techniques and impacts, risks and benefits associated with this area of research. We thought this would be interesting because these kind of technologies keep developing and changing, and the laws and regulations keep being updated both here in New Zealand and around the world. So having some clarity around the terms and definitions could be useful. So thanks again, Revel. Before going much further, I thought it'd be good to hear just a bit about your background, how you came to be somewhat of a national expert around genetic technology.
Oh, thanks, Blue. Yeah, I've been working in this space, in fact, at Plant and Food for the last 17, going on 18 years, and before that, I studied at the University of Auckland. I, since high school, knew that DNA was what I was interested in, and that it formed the blueprint of life as we know it. So I always knew that that was where I was going to end up, and with quite a lot of luck, I did indeed end up exactly here. And so, yeah, I've been spending a lot of time thinking about and actually practicing the idea of genetic engineering or genetic modification, or understanding the DNA that goes into making up plants and animals and people.
And is it correct that you have an advisory role at the moment also?
The other thing I currently hold is a fellowship at the Office of the Prime Minister's Chief Science Advisor, where Juliet Gerrard, who is the Chief Science Advisor, gets me to look into the status of the regulatory systems around the world, because the regulatory systems are trying to keep pace with the changes that scientists are making.
Right, and we'll get into it a bit more later, but did I hear you were the first to use gene editing in New Zealand?
Well, that's where we're going to get into some really interesting ideas about what gene editing is. No, I'm not the first, because while at the moment CRISPR-Cas mediated gene editing is flavour of the month, or flavour of the decade even, actually gene editing has been going on since the 90s. It just used to be very, very difficult, and hardly anybody would do it. What I can claim some credit for is the first flowering plant in New Zealand. I edited a tiny little plant that we use in the laboratory, and so instead of having 4 branches, it had 27.
Very nice. Well, that's where I must have heard it, my role as a molecular biologist in plants. So it was a branching change that you made?
Yeah. I work for a very important person named Kim Snowden, and she is one of the top scientists here at Plant and Food Research. She has been investigating the regulation of branching in plants for the last 30 years. Where I came on board, I edited a gene in Arabidopsis that changed the numbers of branches. It's a very small change, a single base pair, that means that the way the plant develops has been dramatically altered.
And so there's also quite a deep history to the types of genetic technologies that have been around for something like 40 years now. So we're looking forward to discussing that in a bit more depth in the next episode. But just to give us a little bit of a base before we go into some of the terms today, what have the last few developments in genetic technologies been, and what's this most recent step that's different?
So what we as scientists have been developing just recently is a thing called CRISPR-Cas mediated gene editing. It is a precise way of changing a very small number of base pairs in a very large genome. So you might well know that humans have 3 billion bases that make up their genome. In every single cell, there are all of these bases. CRISPR-Cas gene editing enables us to change them one at a time. And so that's very, very exciting. It's very, very precise, and that is a very recent development that we have that developed that level of control.
So the difference between that and previous technology is the precision?
About the precision and about the fact that we influence the existing DNA that is already in the organism. Much of the last 30 years of genetic modification has been adding a gene from somewhere new. So in the simplest case, if we take a bacteria in the lab, and we do this on a pretty routine basis, we can add a small piece of DNA into that bacteria, and we therefore change the way that that bacteria grows. Now that's a genetically modified organism, but it has had to have a whole extra piece of DNA put in. What's new about gene editing is that we are going to only change one base, potentially, of the original organism's DNA.
Right. So many people will have heard some of the following terms that we'll go over, but they might not have necessarily had the definitions before. So I can list some of them off and you can help us explain some of the definitions. So I thought perhaps we could start with the differentiation between traditional plant breeding and molecular breeding.
Right. So traditional plant breeding is something that, in fact, we have been doing for something like 10,000 years. The Mexicans domesticating maize, or the Chinese domesticating rice, the Fertile Crescent producing wheat and all the other grains from that region that we know and love. They did that by taking flowers from one plant and bashing them against the flowers of another plant. And that was a way of making a controlled cross. So now we got a new plant back that was hopefully a little bit better. A lot of them were worse, but we kept the ones that were better. And slowly, keeping the best plant and keeping the best plant and keeping the best plant, then we end up with sweet corn today, long grain rice, sushi rice, wheat that makes lovely, fluffy bread. However, it's very random. We didn't really know what we were doing. So now, as we get to molecular breeding, as we've learned that DNA is the blueprint of life and is present in all of those organisms and controls how they grow, now we look at the DNA and say, well, actually, this is the gene that makes the best bread. This is the gene that makes the best corn. Let's make sure this gene is in the next generation of plants. And by testing for it, by doing various laboratory techniques, then we can make sure that gene is there and we can get a faster method of getting to the right plant in each new generation.
So instead of just picking a plant based on whatever traditional breeding methods, we can use genetic selection to make the cross of the flowers between the plants.
Exactly. With that little bit of extra knowledge, we can speed up the process of traditional breeding enormously.
And so you've mentioned the word genome a few times. I was wondering if you just could quickly define that too.
Right. So the genome of any given organism is the entire complement of DNA that is found in every single cell in that organism, whether that's a bacteria or a plant or a human. In every single cell is a copy of the genome, and that genome is the blueprint, and it tells the organism how to grow.
And so what is genome sequencing?
So in this context, sequencing is reading out the biological molecule of DNA into something that is human readable or machine readable. And the shortcut we use is something you've probably heard of, the bases of DNA, A, G, C, and T. Makes it a lot easier for us to read than the very complex molecule that is DNA. Now, when we talk about sequencing the genome of a whole organism, well, that used to be really hard. In fact, the first genomes we managed to sequence were those of viruses, which only have a few hundreds or thousands of base pairs. But then we moved up to bacteria, and then to yeast, and then in the late 90s we started sequencing the genomes of complex organisms. A small worm that we use in the lab, and then we used the first plant, which is a Arabidopsis thaliana. Now that's quite a big genome, 120 million base pairs, but that's nothing compared to what the human genome of 3 billion base pairs is. And that cost millions of dollars and took many, many years to sequence the first time round. Now, genome sequencing has got to the point that a human genome can be sequenced in about a week for no more than about $1,000. Once we've done the genome sequencing and we've got all the individual pieces of DNA encoded from the chemistry and into the computer, there's another step that needs to happen. DNA is not just a big random shmush in each of our cells. It's carefully organized into chromosomes, a bit like shelving things on a bookcase perhaps. And in the computer, we rebuild those chromosomes into a pseudo or virtual version of the genome.
Right, okay. I like the bookcase analogy where the chromosomes are keeping the genome organized. That's quite a nice analogy. So now that we sort of understand what the genome is, what is genetic modification, and is that different from genetic engineering?
So the difference between the terms you've used there is one that is not really reasonable. Unfortunately, a lot of terms have been mishmashed and stuck together, and sometimes they are used to mean one thing and sometimes they are used to mean another. But fundamentally, what we're talking about is the ability for scientists to move one piece of DNA from that cell over there to this cell over here. Because in this new cell, it will have a function that we can more easily understand or more easily learn about so that we progress the scientific knowledge of the way DNA works.
And so this is easier to do in bacteria because they're more able to accept new genes? Is that?
Bacteria are incredibly easy to make genetically modified, to the point that it takes about 5 minutes for a practiced individual to make a new genetically modified bacteria. We do this in a really safe way because back in the 70s, it was realized that the idea of moving DNA between organisms was a very powerful tool and we'd better be really careful about it. One of the things we do, therefore, is use a system of bacteria that are incredibly weak. We basically keep them on life support just to keep them going at all. And then we help them along a little bit to make sure they live long enough for us to do the experiments we need to. But they literally cannot survive outside of the lab. And that's a feature of our systems we call biological containment.
Another term that we hear a lot is mutation.
So mutation is a totally normal process. And what I normally say when I'm talking to people about mutation and mutants in general is that you'll pretty almost certainly know that you got half of your DNA from your mum and half from your dad, but you're not just the sum of those two parts. There's also this little sprinkling of mutations across your genome that make you 100% unique. Those mutations are what we have been harvesting in our breeding programs. And it's only since we understood what genomes were and that we could sequence them that we would start to find these mutations. They turn out to be very, very common. So mutation is occurring all the time. It's a natural process. And those mutations are what lead to the diversity and variety of life that we see on Earth.
If we think about mutation in the context of CRISPR or gene editing, how are these different? Can you tell the difference?
So mutation in the real world spans an enormous amount and variety of change. Everything from losing a whole chromosome, generally a bad thing, through to just a single base pair changing from an A to a T or a T to a C or something like that. Those smaller changes are the sorts of changes that gene editing can reproduce on demand as opposed to randomly and variously in nature.
And so how is that thought about from a regulatory perspective?
So many of the regulators around the world have started to use terms that let them consider gene-edited changes separately from transgenic changes, because while a transgenic change cannot have occurred naturally, many of the gene-edited changes can, and if they can occur naturally, how would you prove that it occurred naturally, randomly in nature, or precisely and carefully in a laboratory? There is no actual test that can tell the difference between.
Thanks. Now I have some terms around genetic modification that may be a bit more technical, but we'll see how we go. So I've heard of words like knock in and knock out. I wonder if you could explain that.
So scientists like to think they're funny. Don't we all? So Knock in and knock out were terms that were invented back by a series of researchers who were working on mice genetics. And they had a way where they could add a gene, which they would call a knock in, or remove a gene, which they would call knock out. And that's all they mean. They simply mean to add or remove DNA. And because scientists like to share a good joke, we've all started using the same kinds of words. How accurate they are, this is a piece of jargon that the scientists kick around.
Right, so maybe it's a hopeful name. When you do a knock-in experiment, you hope that it actually does put a gene in.
Indeed, in the early days, the success rates were very low.
So I've heard of terms like transgenic and cisgenic as two different types of genetic modification. I wonder if you could explain that.
These types of change are not that easy to quantify, but let's talk about transgenic, as this is what we might call traditional genetic modification. That is when we take a piece of DNA from, let's say, an apple plant and put that into an apricot. That is a transgenic plant because the piece of DNA has moved from one species to another. However, it was thought that perhaps if we took an apple gene from one kind of apple, let's say Royal Gala, and we moved that into a different apple tree, let's say an envy apple, that was a different sort of genetic modification, and that might be cis or same genic. It's a distinction that is potentially relevant in some cases.
It sounds like in the case of genetic editing, this wouldn't really apply because you're not moving a whole gene. Is that right?
And indeed, so for gene editing, these terms don't apply because we're not moving any DNA from anywhere to anywhere else. We're changing the DNA at the site where it normally exists.
Okay, so as far as gene editing, you've mentioned the CRISPR-Cas method. Are there other methods of this precise change, or is this the only way to do it currently?
So CRISPR-Cas gene editing is very new. The Nobel was awarded in 2020. The first publication is in the early teens. But actually, the idea of reaching into a genome and changing a single base has been something that is something of a holy grail for the molecular biology community, the scientists, and has been for a very long time because of the precision that it offers. And over that time, lots of different ways have been postulated or invented to actually do these sorts of changes. But up until the CRISPR method, they were all very, very, very difficult and very, very expensive. And so they were hardly ever done. But it is also why I say I'm not the first person to do gene editing in New Zealand. I think the first gene editing experiment in New Zealand was probably done at Ag Research.
And so in this context, where does cloning fit and what is meant by cloning in plants, for example?
So cloning is a really interesting word that has a lot of pop culture references as well as a technical definition. In fact, several technical definitions. So we are not talking about the idea that we would see like in the film Multiplicity, where we suddenly end up with 4 Michael Keatons. That is purely science fiction and not the way it works. But it is possible to clone a plant simply by taking a cutting off of it and having it produce roots. In fact, if there's any gardeners in the audience, you've probably cloned something yourselves. But in the environment where we are today, we're talking about molecular biology. The cloning we're talking about is to take a piece of DNA from one place and using a laboratory technique, hook it up to a new piece of DNA. And that's about it.
So my early encounter with some sort of cloning in the news was cloning of a sheep that I don't remember how long ago that was.
So that's Dolly. She's a very famous sheep. So that particular type of cloning is yet another technical definition which doesn't actually require genetic modification, in that Dolly's DNA is exactly the same as Dolly's mother's DNA or her clone's DNA. So while the technique does require a laboratory to produce a cloned Dolly the sheep, it does not actually change the DNA directly.
Okay, so it sounds like cloning can happen at an individual level, but you can also clone a gene, and so it happens at a few levels.
It does happen at a lot of levels, and that's why I think you have to be quite careful with that term in particular, because you have to make sure you're in the same context as somebody, because if you're talking to a gardener about cloning their favorite rose, you're talking about a completely different thing than somebody talking about cloning the latest, greatest gene for baldness and putting it into mice and they become bald. There's a very, very different. Make sure that you're in the right space when you are using that
Quite
word.
different processes, yeah. So we've discussed about all these various techniques. How are they currently used?
Well, I use them on a very routine basis in the lab, and there are many scientists around New Zealand who use them routinely, day in and day out. We use them in containment laboratories and containment glasshouses. These are very strictly regulated environments which are a physical containment mechanism to make sure that nothing gets out.
And so currently it's just a research tool?
Exactly. There is no cultivated, genetically modified crops or animals grown in New Zealand.
And so what about any potential risks?
There are absolutely scientific risks involved in the use of these technologies. That is why we have been experimenting in the lab and in containment glasshouses now for 30 years. Each new change needs to be assessed for, did the change occur as we planned? Did that change the plant in the way we intended it to? Did it do anything we didn't intend it to? That is why we still have quite a long process following the genetic modification of any plant through testing in glasshouses, testing in field trials before any release into commercial production.
So if we just move a little bit into legal definitions around how these types of technologies are used, I know different countries approach this different ways. I wonder if you could just update us in the New Zealand context.
So New Zealand's regulatory environment has not changed significantly since the rules were written back in the early 2000s, and even then that was only a minor rewrite of a rule written in 1996, which is called the Hasno Act. So we have a definition of genetic modification in law that is already 30 years out of date. However, some things haven't really changed. It is still the movement of a piece of DNA from one organism to another. But when the rules were written, we didn't know nearly what we know now. 30 years of progress is a long time. So the laws in New Zealand are simply out of date. On the other hand, many countries around the world, Australia, Argentina, Brazil, Canada, and the USA have had updates to their rules relatively recently, and they continue to evolve. The most recent changes are that of the UK, which recently had a very significant rewrite of the way they regulate genetic modification. And the one that's in progress right now is the European Union, who are having an even more dramatic change to their rules around genetic modification.
A recent news story I saw in New Zealand was around a type of genetic change called a null segregant. I was wondering if you could explain that for us.
Well, I would direct you to a lovely video produced by Plant and Food Research that will explain it better than I can in words, but let's look at it this way. When we make a genetically modified organism, we put a single copy of that gene into the plant, and we can grow that plant up. Let's say it's a tamarillo, and that plant is quite clearly genetically modified, and it has the transgene in it. However, for those of you who've ever tried to grow tamarillo, you'll know that they make plenty of seeds. And you can collect the seeds from your tamarillo, and you can grow new tamarillo plants. Of the seeds that you get back, only 3/4 of them will have the transgene. The other quarter will not. And those ones are called null segregants. So they've gone back to being non-transgenic. And that was carefully restated by the New Zealand EPA in just the last few months.
So it sounds like it's the situation where one of your parents is genetically modified, but you did not receive that from that parent, so you don't have that change.
Yep, that child would be a null segregant as well.
And so how does New Zealand currently treat plants, for example, that are null segregants?
So that's what's changed. And actually, it was quite a surprising decision from the EPA to rule that null segregants would be treated as non-genetically modified. Up until this new ruling, it had been thought that the wording of the Has No Act, again written in 1996, actually said that even those 1/4 of Tamarillo plants which don't actually have the transgene would still somehow be genetically modified, even though they didn't have any of the transgenic DNA.
So in the context of these definitions, if you were sort of to look out into the future and make a few educated guesses based on your expertise, if this technology matures, how could it go right or wrong in the context of plants and, say, humans?
So I'll stick to talking about plants because I am a plant biologist. So with the oncoming climate change that will absolutely impact the way we can and will grow plants in New Zealand in the future, we need every tool in the toolbox to be able to get those plants to a state where they are still productive and they are making fruit, making wheat, grains, whatever, even as the climate changes. That, I think, is probably the place where I think gene editing will be accepted and acceptable and deployed in New Zealand, because that is where we need to focus our efforts going forward. If we're going to stay a country that makes a huge proportion of its income from growing food, we have to be able to grow food even in the new climates that we will see going forward. So those sorts of gene editing changes, we are already looking at ways to make sure that apple trees, which need a nice cold winter before they produce fruit in spring, can continue to do that even if the winters aren't so cold.
Okay, great. Thank you very much, Revel. That was very insightful. And we look forward to our next discussion soon, where we'll go deeper into some of the new technologies, where and how they're currently being used overseas, what products are coming online, and how other countries are developing a framework around the utilization of these new technologies. Until then, thank you to our listeners. Please subscribe or follow Scigest on your favorite platform, and we shall see you next time.